Assalamu alaikum. I'm Professor Dr. Haidar Jawad Mubarak. This is a presentation to the topic of comparative anatomy of the cardiovascular system. The primitive heart is formed just like a tube, and therefore this heart, or the primitive form of the heart, is called a single circuit heart because it is just like a single tube. This single circuit is usually divided into four chambers, or containing four chambers, which are from anterior to posterior: the conus, then the ventricle, then an atrium, then the sinus venosus. Of course, the venous blood reaches the heart via the horns of sinus venosus, and from the sinus venosus the venous blood reaches the atrium, the single atrium, that pushes the blood toward the ventricle, and the ventricle pushes the blood toward the last part of the single circuit or single tube heart, which is the conus, that represent the outflow of the single circuit heart, and from this conus to the aorta, to the blood vessels. Such a primitive form of heart is also seen in cartilaginous fish and also seen in bony fish, but here the conus is replaced by another structure which is called bulbous arteriosus. In a more developed, functionally developed heart, we have two atria instead of one in the primitive heart. The primitive single circuit or tube-like heart contain one atrium, but a more developed heart structure contains two atria separated by a septum with a single ventricle, and therefore this heart contains three chambers with two atria and one ventricle. Here also we can see number 13, which is the conus, but actually this is number 9, indicated the conus, which is here demonstrated by the blood vessels derived from the conus. And this is a more developed structure of the heart, which is seen nowadays in amphibia as a frog. So the heart of fish, as we had just shown, is formed of a single circuit or a tube-like heart with four dilatations or chambers. In amphibia, it shows two atria separated by a septum and having a one ventricle. In the more developed, the heart that has a functionally developed structure more than that of the three chambers seen in amphibia is seen in amniotes, the animals that shows amniotic membrane during fetal or during embryonic life. Such amniotes will have a septum not only in between the two atria but also inside the ventricles, and thus this more developed, more functionally developed heart has two atria and two ventricles. Sometimes the septum are incompletely dividing the atria and the ventricle, just like in reptile. Others show complete separation of the atria and complete separation of the ventricles by an intact septum, just like in bird and mammal, including, of course, mammal including a human. So the human has a complete septum, intact septum, separating two atria and two ventricle, and this is the very well developed functionally heart. As we said before, that phylogeny recapitulate ontogeny recapitulate phylogeny. So during ontogeny of the heart, of course, the term ontogeny means development of an organ in various stages of pregnancy, in various stages of embryonic life. So if you look to the heart stages during development of embryo, you can see that the earlier stages of the heart in a human is also single circuit or single tube heart, just like that seen in fish, which is called single circuit heart. It is formed of sinus venosus, single atrium, single ventricle, and bulbous chordates, just like that's seen in fish. So in later stages of development, the human embryo heart will show changes that shows folding and septum formation to form the final configuration of the heart. And therefore you can see that the poor developed heart is seen in earlier stages of the heart in embryo, and the more advanced or well-developed functional heart is seen in the full-term developed fetus or developed human at the end of pregnancy. Of course, due to that principle that ontogeny recapitulate phylogeny, which I had just mentioned now, we may see deviation from normal ontogeny. For example, we said that some of the septum in reptile are incomplete septum. Before a while we said that the septum may be between the atria and the ventricles are incomplete, and therefore we may see a defect in the atrial septum or defect in the ventricular septum in human due to the same reason which we are considering it as a principle that ontogeny recapitulate phylogeny. This is all about the comparative anatomy of the heart. We said that our topic today is cardiovascular comparative anatomy. So the cardiac comparative anatomy had been described, and now we may pass to the description of the vascular comparative anatomy. This figure shows you the primitive pattern of arterial channels in the body. It shows this is the heart tube, which is a primitive heart tube, a single circuit or single tube heart tube, and the vessels here are also primitive, just like the heart shown in this embryo, of course. It shows a ventral aorta projecting out of the conus cordis or out of the bulbus of the heart from the anterior aspect of the single circuit heart, the ventral aorta. And we have two dorsal aorta. The ventral aorta is ventral to the pharynx and gut, while the dorsal aorta, the two dorsal aorta, are dorsal to the pharynx and gut. And anteriorly we can see that the dorsal aorta are connected to the ventral aorta by 6 arteries, which are called aortic arches. These are, of course, 6 pairs of arteries, because we have two dorsal aorta, and therefore we have a pair of dorsal aorta and pair of aortic arches, right and left. So this is the primitive pattern of arterial channel. From this primitive pattern, there will be a more developed configuration. Actually, the more developed configuration will show two circulations, which are the smaller pulmonary circulation and the larger systemic circulation. In each circulation, there will be a vessel that anastomoses in a terminal branches with the venules that returning the blood to the heart. And such an anastomosis is seen in the lung, in the pulmonary circulation, but are seen in the upper body, in the lower body, in the kidney, and even in the liver and GIT, gastrointestinal tract. So this is the more developed arterial channel. It forms pulmonary circulation and systemic circulation that branches profusely to return the blood to the venous side. Actually, the developed blood channels or arterial channels show a system which is called portal system. You will always hear in your learning of anatomy, you will hear a portal system. So what is the portal system? Portal system is a term indicating a system containing two series of capillaries. What is meant by that? You can see, for example, here the blood from the artery goes to the gut, to the intestine, for example. And at the intestine there will be capillaries. Here there will be absorption of food toward these capillaries. The veins getting out from the intestine will form a vein which is called portal vein, that will form another capillary system in the liver. And from the capillary system of the liver, the food that had been digested from, absorbed from the intestine is now metabolized in the liver. And after this capillary system in the liver, the blood returns via the hepatic veins to the heart and inferior vena cava. So because we have two capillary systems that are unconnected, capillary system in the intestine and capillary system in the liver, so this is a portal system. Again, you may see here a portal system in the kidney, capillaries in the lower part of the body and then connected with capillaries in the kidney. So this is a portal system. In the brain, we have a gland which is called hypothalamus, and this hypothalamus connected with another gland which is called pituitary, and each of these containing a plexus of capillaries, and so it is hypophyseal portal system from the hypothalamus capillaries to the pituitary capillaries. Of course, large capillaries in the hypothalamus are called sinusoids because the diameter of this capillary are slightly larger than any other capillaries. That's all about comparative anatomy of the heart and arteries. The 6 aortic arches that we had just described in the primitive configuration of the arteries. We said we have 6 pairs of aortic arches connecting the bilateral dorsal aorta with the ventral aorta. These 6 arches are equipped in each species of animal to produce the final normal configuration of arterial system. Some of these 6 arches will disappear, the other will be further developed, resulting in the specific configuration for each species of arterial system. And you can see, for example, in bird, these are the gray drawn arches are arches that disappear, and the colored are arches that developed. In birds, others are disappeared and others are developed, and so on. In each type of animal, some of these 6 aortic arches will disappear, the other will further develop, resulting in a specific configuration of the arterial system. If you look to the human, these are the dotted line representing the 6 aortic arches connecting the dorsal aorta with the ventral aorta. Some of these, as I said, will disappear, the other will well develop to form this final configuration of the great vessels in the chest. And of course, you will study how this configuration of the 6 aortic arches resulting in this configuration of the great vessels connected with the heart in your course of embryology at the second year of medical school. Venous system is rather simpler in its description regarding the comparative anatomy. Veins in the body in a primitive manner contains cardinal veins that drains the body of the embryo, which are anterior cardinal and posterior cardinal, draining the anterior cardinal draining the anterior part of the body, while the posterior cardinal draining the posterior part of the body. And also we have renal veins draining the kidney, or they are called subcardinal veins. And we have vitelline veins that draining the intestine and associated with the liver circulation, and coronary veins draining the heart. The more developed configuration of the venous drainage and functionally more developed venous drainage may be seen in higher vertebrates that they contains pulmonary veins in addition to these veins, and a vena cava. This is simply about the configuration of the comparative anatomy of the venous channels in a developed and early developed, poorly developed and well developed vertebrates. The configuration, as I said, of these venous channels will show disappearance of some of the veins and progressive development of the other veins, resulting in the specific pattern of venous channel in each species, and this is the venous pattern seen in a human, and you will learn that later on. That's all about comparative anatomy regarding cardiovascular system. Of course, I'm considering a summary to the subject. The subject is carrying much more information than that, but I'm taking things according to the simplest way. Thank you very much.